All right, so today is our second day of our final review for the AP Bio exam. This is Unit 2, Review Cells. The first thing I'd like to do is talk about microscopy. Microscopy, not microscopy. Microscopy. Whoa, what's going on? Okay. This is just the study of cells using or by using microscopes. All right, when it comes to microscopes, there's three different types that we need to know about for this class. Number one is a compound light microscope. This uses a series of lenses and light to magnify the specimen and to make it visible. Now, the different types of magnification are called objectives. And when you're using a compound light microscope, you have the 4 times objective, the 10 times objective, the 40 times objective. When you guys go to college, you'll have a fourth one called the 100 times objective. These objectives are just like the different strengths of magnification. So if I say 4 times, you're looking at something at 4 times normal magnification, 10 times normal magnification, and so on and so on. These are called objectives. These are the types of microscopes that we have here at school. Most high schools have plenty of compound microscopes or compound light microscopes. The other 2 types of microscopes are called electron microscopes. And these are enormous machines. They take up a huge room. They're not like something you can hold in your hand. Electron microscopes can see things greater than 2,000 times magnification. That's crazy. That's really, really strong. That's very, very small. What they do is that they shoot a beam of electrons at a specimen. Now, because this is shooting electrons at the specimen must stay still. Now, also because you're shooting a beam of electrons at the specimen it needs to be dead. It cannot be moving. And even if you had it like paralyzed, shooting the electrons at it would kill it. And so the specimen needs to be dead. Being dead will ensure that it's not going to be hurt and that it will not move. So the second type of electron microscope is called a scanning electron microscope. It is abbreviated as SEM. And what this does is it coats the specimen in electrons. So you can actually see the surface of it. If you've ever seen images of scanning electron microscopes, they can actually look pretty scary. People take scanning electron microscope images of insects or like larvae and worms and stuff like that, and it makes them look so huge and scary when it's something that's so small. They're very detailed. The electrons just cover the surface of it. And the third type is called a transmission electron microscope. Or TEM. Okay? Rather than coat the specimen with electrons, a beam of electrons goes through it. It goes through the specimen. And so you can actually get a layer by layer image of what's being seen. Now, I do want to indicate to you guys that these types of microscopes are better suited for viewing organelles. While a compound light microscope is better for viewing cells. If we were in a regular classroom and if it was a regular kind of school year, we would have done a lab where you guys would have a toothpick and you would scrape the inside of your cheek with the toothpick, and that would collect a bunch of cells. We would then stain those cells with a purple dye, and you could actually look at your own cells underneath the microscope. You would be able to see your plasma membrane, your cytoplasm, and your nucleus. You could see all these different cells. Things that you would not be able to see would be things like mitochondria, ER, Golgi, stuff like that. Those are way too small. You need an electron microscope to see that. All right, let's move on. Let's go over here. We're going to do types of cells. You don't need to make this indentation like I just did. I just have a bunch of other notes over here on the left, so that's why I'm scooting over here to the right. What were the first types of cells on Earth called? Prokaryotic cells. These are prokaryotes. They're the first cells on Earth. They evolved about 3.6 billion years ago. BYA. Let's see. They are single cell. They're unicellular. Unicellular means single cell. They reproduce asexually. I want you guys to really realize how asexual reproduction works. Penises and vaginas are billions, if not trillions of cells. They're organs. They're body parts. We're talking about an organism that is one cell. So if you're thinking of sexual reproductive organs that are billions to trillions of cells versus one cell here, this little cell is not going to be reproducing sexually. It only is one cell. Here's something that might blow your mind. A female's egg cell is the biggest cell of the human body. You could actually see it without a microscope. You'd have to be really close, but you could probably see it with the naked eye. That is hundreds of times larger than a prokaryote. So if you think about that, an egg cell, an egg cell comes from an ovary, and then it goes to the uterus, and sperm has to get there through the vagina to the uterus, all this stuff. What I'm trying to really convey to you all is you can't be reproducing sexually if you're made up of just one cell. The types of organisms that are prokaryotes are bacteria. Yes, Ella?
How do they evolve then if they're only producing asexually?
They form colonies, which leads to multicellular organisms. They form little rafts of multiple cells working together, and that will make one organism. That's multicellular life.
They just combine themselves?
Yep, and over time they would become one thing. So bacteria, whether they're good bacteria or bad bacteria, like good bacteria could be the bacteria in your intestines, E. coli, that's good. Then you got bad bacteria like syphilis or flesh-eating bacteria, necrotizing fasciitis. One second, let's pause this. Okay, sorry guys, I got a phone call, so let's just get back to it. The types of the domains that are made up of bacteria is, hold on, screen froze a little bit. There we go. Now we're back. The domains that include prokaryotes is domain Archaea. Archaeans are going to be extremophiles. Extremophiles are organisms that like to live near hydrothermal vents, volcanoes, sulfur springs, salt lakes, stuff like that. And the other one is going to be domain bacteria. Let's see what else I want to include with this one. Hold on a second. Where did I put my cards? Oh, there they are. All right. We're going to go over a whole list of all the different parts of the cell, and I'm going to tell you if it's a prokaryote or a eukaryote. Oh, here's one. They have circular DNA. When I say circular DNA, it looks like this. All right. And let's go over here to eukaryotes. Eukaryotes evolved about 1 billion years ago. They can be unicellular or multicellular. You guys are all multicellular organisms. If they're unicellular, they're typically going to reproduce asexually. But if they're multicellular, and it could be asexual or sexual. It depends on how many cells there are. Let me ask you guys this. If I had an organism that was 10 cells, do you think it reproduces sexually? No, it's still going to be asexual. But if you have a more complex organism, then that's more likely to be sexually reproducing. The organisms that are eukaryotes would be protists. Do not get protist confused with prokaryote. We have fungi, we have plants, and we have animals. All of these are eukaryotes. And they have linear DNA. All right. Okay, so what I would like to do now is I want to make this big, pretty chart of the different organelles and other structures found in a cell. We're going to identify if it's found in a prokaryote, a eukaryote, either one or the other or both or none. And then we'll get into what the actual structure is, okay? So let's make this chart. This is going to go away, so just be prepared for that. This is going to be a pretty big chart. Yeah. So I'm going to have the structure. We're going to have prokaryote. We're going to have eukaryote. And then we'll have a description. Okay, you ready? Here's the first one I'd like to do. Cell wall. Otis, are these found in prokaryotes? No. Yes, they are. Emma, are they found in eukaryotes? Yes. Yes, they are. Cell wall is basically protection, everybody. Now, here's a little pop quiz. We did this one yesterday. What is the cell wall of a bacteria made of? No, not chitin. Peptidoglycan. 9 days until the exam. What is the cell wall of a plant made of? Cellulose. What is the cell wall of a fungi or a fungal cell made of? Chitin. How about an animal cell wall? Trick question. They don't have one. Animals do not have a cell wall. Okay? So you kind of see how this chart's going to go? Let's do a plasma membrane. This is going to be a sizable chart. There's a lot of things to go over. Do prokaryotes have a plasma membrane? Yes. How do they keep things in and out? Yes, they do. Do eukaryotes have one? Yes. The plasma membrane is a semi-permeable membrane. This means that it allows some things through and other things cannot go through. What types of things can diffuse right through the membrane? Polar or nonpolar? So this means that the membrane is actually nonpolar. The membrane is composed of a phospholipid bilayer. The plasma membrane has channel proteins scattered throughout it, and it also has cholesterol. Whenever I think of a plasma membrane, I like to think that it resembles the surface of Tampa Bay, where you see the waves are generally rocking back and forth, nothing major, not like a hurricane, just up and down, really easy, really nice. And if you put a beach ball in the water, the beach ball is going to move around because the water is moving around. That's all due to cholesterol. Cholesterol is responsible for the fluid mosaic model. This simply means that parts that are embedded in the membrane are able to move around. If it weren't for the cholesterol, it wouldn't be able to do this. Okay. Let's get to some other structures. Let's start from the inside and we'll work our way out now. Let's do the nucleus. Emma, do prokaryotes have a nucleus?
No.
They do not. Do eukaryotes?
Yes.
Yes, they do. The nucleus is where DNA is stored. That DNA can either be chromatin, in which it is decondensed, or it could be chromosomes, in which it can be condensed. Do either of you know what that little structure inside of a nucleus is called? The what? That's not the whole name. The nucleolus. Now think about this. This nucleolus is found inside the nucleus. So do prokaryotes have one? No. Do eukaryotes have one? Yes. The nucleolus is inside the nucleus. This is where ribosomes are assembled. It's basically where ribosomes are made. How about the nucleoid? Do prokaryotes have this? Yes. Do eukaryotes have this? No. So in my experience, a lot of students get the words nucleolus and nucleoid confused. Don't do that. The nucleoid is not a structure. It is a region. It's an area where DNA is found in a prokaryote. If you've ever been in an airplane and you've flown through a cloud, the airplane didn't have to go through a wall. It just goes through the cloud. That's kind of like what a nucleoid is. It's just an area inside the cell of a prokaryote where you would typically find the DNA. Okay? Okay. Let's get to the next one. Ribosomes. Otis, do these have, are these found in prokaryotes? Yes. Emma, are they found in eukaryotes? Yes, they're found in both. In my experience, a lot of students think that ribosomes are organelles, and therefore they know prokaryotes don't have organelles. Every kid knows that. But they mistakenly think a ribosome is an organelle, so they think, okay, prokaryotes can't have those because they can't have organelles. A ribosome is not an organelle. It's just basically ribosomal RNA. When I say ribosome, you say protein. That's right. This is the site of protein synthesis. This is where proteins are assembled. Here's a question that you won't get until next Tuesday. What do you call that process of making a protein from RNA? What's that known as? Translation. That takes place at the ribosome. Okay, let's keep going. The endoplasmic reticulum. Otis, do prokaryotes have this? No. Emma, do eukaryotes have this? Yes, it is an organelle. So this is an organelle that is a series of membranes and sacs. There are two types. There's a rough ER. What is a rough ER covered in? They call it rough. It's covered in ribosomes. Because of the ribosomes, the rough ER makes protein, but just by itself, the ER makes lipids. So the rough ER makes proteins and lipids. The smooth ER just makes lipids. This is responsible for producing hormones, like steroids. Steroids are lipid-based hormones. Testosterone, for example. And they help to detoxify. So let's see if you guys can figure this out. If the smooth ER can really help to detoxify your whole system, what organ would you expect to have cells with a lot of smooth ER in it? Liver cells. That's right. Very good. The liver cells are called hepatocytes. Hepa means liver. I'm sure you've heard of hepatitis. Hepatitis is a liver disease. Well, liver cells have a lot of smooth ER because it helps to detoxify your system from things like drugs and alcohol and other crap. Okay, let's go to the next one. Let's do Golgi, the Golgi body, sometimes called the Golgi Apparatus. Otis, is this found in a prokaryote? No. Emma, eukaryote? Yes. This is called the post office of the cell. It receives packages from the ER. It sends packages to the plasma membrane. Otis, I want to ask you a question. If the packaging that a Golgi makes can easily blend into the plasma membrane, what must the packaging be made of? What is the plasma membrane made of? It's made of a phospholipid bilayer too. These little packages I keep referencing are called vesicles. They're like little bubbles that allow for the transport of different contents like proteins. All right, let's keep going. I told you this is going to be a big chart. We have lysosomes. Otis, are these found in prokaryotes? No. Emma, eukaryotes? Yes. Now, this comes with an asterisk. This is animal cells only. You will not find these in fungi or plants. They break down unwanted material. They use digestive enzymes or hydrolytic enzymes. What type of cells in your body would you expect to have a lot of lysosomes, knowing the fact that lysosomes break down unwanted things? No? What cells in your body look for things that shouldn't be in your body and get rid of them? White blood cells. You have a lot of lysosomes in white blood cells. Okay, next one we didn't really talk about in class. They're called peroxisomes. Prokaryotes do not have them. Eukaryotes do. You guys know that little brown bottle that has hydrogen peroxide? Hydrogen peroxide is H2O2. Well, hydrogen peroxide is a byproduct of cell respiration. It's really not that good. It's very toxic. And so to protect your cells from this, the peroxisomes convert H2O2 into harmless water and oxygen. So when you're going through cell respiration, your cells will produce hydrogen peroxide, and that can be very dangerous in high concentrations. So the peroxisomes are going to be little vesicles that contain peroxidase, an enzyme that breaks down hydrogen peroxide into plain old water and oxygen. All right, we're getting close to the end. We have a few more. We have the mitochondria. This is a big one. Otis, are these found in prokaryotes? No. Emma, how about eukaryotes? Yes. Here's what I like to say, Otis. I kind of got from your voice. You're like, no? You weren't sure. It's an organelle. Do prokaryotes have organelles? No. Here's why I also like to say, prokaryotes are ribosomes. Well, I said that really bad. Prokaryotes used to be in the mitochondria, or the mitochondria used to be the prokaryotes. The mitochondria used to be its own living thing. This is the site of cell respiration. It has its own DNA. Whose DNA is in it? You remember that? Mom's DNA is in the mitochondria. It has a double membrane. And what is that idea, that theory that describes the origin of the mitochondria? Do you recall what that is? Endosymbiosis. That's right. This is the result of endosymbiosis, where bigger cells absorb smaller cells, and they became one. All right. Let's see what else. They behave independently of the cell. Remember, they used to be their own living thing. Oh, one other thing I forgot to mention. Found in all eukaryotes. Plants, fungi, animal, protists, you name it. Let's go to chloroplasts. Otis, prokaryotes? No. Emma, eukaryotes? Yes. Whoops, wrong color. Okay. This is the site of photosynthesis. There's going to be a lot of similarities here with the mitochondria. But because it does photosynthesis, what's the only cell it's found in? Only in plants. You're not going to find it in fungi, animals, or protists. Some protists. It has its own DNA. Again, you're going to see a lot of same things with the mitochondria. Double membrane. It is a result of endosymbiosis. And they behave independently of the cell. All right. Should be pretty downhill. I think we maybe have 4 more, maybe less. We have a vacuole. These are not found in prokaryotes. They are found in eukaryotes. This definition is very easy. Storage. This is where cells can store water and food. It's mostly in plant cells, but I can't say it's not in other cells. But you would find them in plant cells more than any other type of cell. It stores water. It stores food. In fact, a plant cell can be 90% vacuole, and the vacuole holds water. We all know how important water is to plants. Okay, we have cilia and flagella. They are found in prokaryotes and they are found in eukaryotes. It's basically meant for movement. If you guys can look at the screen, cilia looks like this. Here's a cell, and cilia looks like a bunch of little oars, like from a Viking ship, where they're all paddling in the same direction. While a flagella, and I'm sure you've seen this like on a sperm cell or something, it's a cell with a little tail. Now, what I just drew here is a sperm cell. The sperm cell is the only human cell that has a flagella, but if you had a bacteria like this, some bacteria can have up to 3 flagella. The sperm cell swims like an alligator. It just flops its tail back and forth, back and forth. While prokaryotes that have 3 flagella, they actually spin them like the blades of a boat motor. All right, and I believe this is the last one, cytoskeleton. There is some research that some scientists say they found it in a prokaryote, but for now we're going to say no because it's still early research. It is found in, wrong color. It is found in animal cells, or excuse me, eukaryotic cells, but I do want to indicate that it's animals only. It provides the cell with shape, structure, and support. I want to see if either of you can tell me. I just told you that only animal cells have this. Why don't plant cells and fungal cells have these? What do they have instead to give them shape, structure, and support? The cell wall. The cell wall gives them that. Animal cells don't have a cell wall, so they need something else to reinforce them and to give them structure. All right, guys, there's our big chart. So we are going to now switch over to diffusion, okay? So let's do it. Our chart is finished. Now let's get to diffusion. Diffusion is simply the movement of particles across a membrane. Actually, no, excuse me, across from region to region. There are 2 types of diffusion. The first one is called passive transport. Just by the name, guys, does this require energy? No, you can tell by the name. I remember when I was in school, it's like that's passive. You don't need energy to just let something pass by. This requires no ATP. And in what way do particles diffuse? From high to low or low to high? High concentration to low concentration. That's right. Okay. There are 3 types of passive transport that we learned in this class. First one is simple diffusion. An example that I like to do in class is I would stand in the corner of the class and spray someone's Axe or perfume or something that is very strong smelling, and I would spray it in one corner of the room. We would describe that corner as being a high concentration of whatever aroma that is. And then over the course of maybe 1 minute, the whole room fills out with that smell. That is simple diffusion. If your mom is cooking something that's very smelly, like in a good way or even a bad way, in the kitchen, eventually the whole house will smell like it. That's diffusion. Diffusion works with sound, it works with smell, it works with light. It goes from high to low. Simple diffusion is just small molecules going from one region to the next. The next is facilitated diffusion. Facilitated diffusion is passive transport. It uses channels to transport large molecules. And if we're talking about the plasma membrane, those molecules will also be polar. Can a polar molecule pass through the plasma membrane without help? Yes or no? Can a polar molecule pass across the membrane without help? No, because the membrane is nonpolar, so you need a channel. Number 3 is osmosis, which I'm going to spend a few minutes on here. We have about 12 minutes left in this video. Osmosis is the diffusion of water. Now, I want to really focus on this, and I need you guys to really listen up. This is the last time I can explain this to you. There are 2 different ways to try to determine which way water will diffuse. I'm going to teach you those 2 different ways, but you still end up with the same result. So here I have a semi-permeable membrane. And here's my solvent. And let's say on the left-hand side, I have 0.6 molar. Let's say on the right side, I have 2.2 molar. Otis, which side is more concentrated? The right side. Clearly, it has more solute. So one way to view this, here's the first way. I told you that I'm going to teach you 2 ways. Number 1, water diffuses towards a greater solute concentration. So Otis, based on this, which way will water diffuse? Left to right or right to left? You would be correct. Left to right. Last time I checked, 0.6 is less than 2.2, or 2.2 is more than 0.6, so water will diffuse towards the 2.2. So that's one way to figure out which way water is going to flow. Here's number 2. Emma, I want you to listen very carefully. You might have to think about this for a second. Emma, which side has less water? The left side or the right side? That's right. If the right side is 2.2 compared to 0.6, that is the amount of solute. So here's what I hope was going through Emma's head. The right side has more solute, so it has less water. So water will just go from high to low. Water diffuses towards a lesser water concentration. And Emma, which side did you say has less water, the left or the right? The right. So which way will water diffuse, left to right or right to left? Left to right. That is correct. See, it's the same outcome, and there's just 2 different ways to explain it. Good job. Okay. Let's talk about hyper, hypo, and isotonic. Otis, I have a cup here. I have a solution inside, and I have a cell. Did you take the second mock exam? No. Well, do you remember when we were reviewing it last week, how about that problem with the grapes and peeling the grapes and putting in soda and stuff like that? Well, that whole example is based on hyper, hypo, and isotonic. That's all about water diffusion. Otis, let's say that this cell, or grape, or whatever you want to call it, has a molarity of 0.3, and the solution is 0.7 molar. Which way is water going to flow? Into the solution or into the cell? Into the solution. So water is going to leave the cell, right? What type of solution is this? Is the solution hypertonic, hypotonic, or isotonic? And here's why I'll hint it for you. Hyper, he just said hyper. Hyper means too much, right? Like, oh, you're really hyperactive. That means too much. The solution has more than the cell. That's why we call it hyper. This is a hypertonic solution. And what would happen to the cell? Would it shrivel, swell, or stay the same? The cell will shrivel. Okay, let's do another one. Emma, your turn. We have a cell over here. It's 0.3 molar. Let's say the solution is 0.1 molar. Which way is water going to flow? Into or out of the cell? Into the cell. Because it always goes towards more concentration. So what type of solution is this? Hypo. My first year teaching, I had a student say hypo like a hippo. So what's going to happen to the cell? It's going to swell up. Okay, and for the last one, either of you can say what's going to happen. Let's say we have a cell that's 0.3 molar, and the solution is 0.3 molar. So they're both 0.3. Which way will water flow? It's going to be even. This is called isotonic. However much goes in, goes out. And in this one, the cell is unchanged. Got it? That's hyper, hypo, and isotonic. All right, we have a few more topics. We have a few more minutes. The next thing I want to talk about is water potential. You guys have this equation on your AP Bio formula sheet that you'll get during the exam. The equation is this. Water potential equals solids potential plus pressure potential. This is water potential. I'm going to try to pick up the pace here. We're running low on time. Psi S is solute potential. And psi P is pressure potential. Now, I want to clarify a few things here. If I say I had fresh water, are there any solutes in that water? No. So fresh water's water potential, or they might call it pure water, that is going to equal 0. Pure water or fresh water has a water potential of 0. I want you guys to look at solute potential. Solute potential has its own equation, negative I CRT. I stands for ionization constant. C stands for concentration, which is also molarity. R is just a constant for water potential. You'll be given that. And T is temp. But what I really want to focus on is I. In my personal opinion, I think that chemistry should be a prereq for this class, but they don't ask me. But here's why I think it should be. I want to see if you guys can tell me what is the ionization constant of a few solutes, and I'm going to tell you why they are. So let's do the first one, guys. NaCl. If I place that into water, how many ions will it fall apart into or ionize into? 2. Sodium and chloride. Its ionization constant is 2. How about magnesium chloride, MgCl2? How many would that be? 3 magnesium and 3 chlorides, or 2 chlorides. Okay. How about AlF3? How many would that be? Oops, one extra line. That would be 4. How about Al2O3? 5. Okay, last one. Sucrose. C12H22O11. Nope. 1. You know why? It's molecular. Sugar is molecular. When you place it into water, it doesn't ionize. That's a chemistry problem. If you see a giant molecule like that, sucrose, glucose, or any type of carbohydrate, sugar, when you place it into water, it doesn't fall apart. It stays together as one. And so when it stays together as one, its ionization constant is 1. Got it? One thing I do want to say with pressure potential, just a little fun fact, keep this in mind. An open container would be 0. So if they mention to you in an FRQ, you have an open container, that should be like a red alert. It's 0. All right, we're almost done. We have less than 2 minutes, so I'm going to try to hustle through this. Active transport. Does it require ATP? Yes. It does require ATP. It goes from low concentration to high concentration. So something that's a high concentration will be made even higher. It uses pumps in order to achieve this. You have to power a pump. ATP powers those pumps. Okay, I think we're barely going to make it. Second to last thing I have for you guys is apoptosis. Can you define apoptosis for me? Programmed cell death. Every 3 seconds, 40,000 of your cells go through apoptosis, but they are replaced by mitosis. Here's my last part for you, plasma membrane. If the bell rings, just stick around for 30 seconds if you can. I want you to draw a brief portion of a plasma membrane. Okay, what region is nonpolar, Otis? The inside or the outside?
Inside.
The inside, good. And that means the outside is polar. Here's my last question for you all. Why do the polar regions face outward?
Because they're hydrophilic.
Yeah, but you're right, but that's not telling me why. You're telling me a fact. I want to know the reasoning. Why are the polar regions facing outward? Well, what do you call the outside of a cell? It's called the extracellular matrix. What do you call the inside of a cell? The cytoplasm.
Oh.
What are the polarities of the extracellular matrix in the cytoplasm if the polar regions are facing them? They're also polar. You want polar to face polar, and you want nonpolar to face nonpolar. Let me tell you a quick question before you go that I remember I got on a test in college. You ready? Here's how it goes. You go to an alien planet, and the oceans of this alien planet are hydrophobic. You find a sea creature in this ocean, and you dissect it, and you find out that its cytoplasm is also hydrophobic. What do its plasma membranes look like? It's flipped. That's right. It's reversed to where the nonpolar tails are facing outward like this, and the polar regions are facing inward because you want polar to face polar and nonpolar to face nonpolar. That's it.